Good afternoon, everyone. Thanks for inviting me. Thanks for the organizers for inviting me, especially Ilya. And today I'm going to share with you what we do at Renewal Bio and how we're developing technologies for rejuvenation. So at Renewal Bio, what we do is basically harnessing the full potential of pluripotent stem cells. We've developed a very cool platform to differentiate pluripotent stem cells. So I would like to start with this painting by Lucas Cranach the Elder, where old people come from one side of the fountain of youth and come out young. And of course, unfortunately, we haven't found a fountain of youth yet as far as I know. But it would be cool if we, much like cars, can basically change parts that are old and rejuvenate them by newer parts. And for that, we've developed a new entity, and we call it Stembroids. It's a cell therapy platform that we use to differentiate pluripotent stem cells, and we basically use the environment or the microenvironment or bioenvironment that support differentiation properly that happens in real life, just with cells, with pluripotent stem cells, and that's how we're doing it. As you know, there is an immense need for cells for a lot of different conditions. However, it's not really available. There are a lot of reports in the literature that fetal tissue can really rejuvenate conditions or aging organs or aging tissues. However, it's ethically controversial. We can't really harvest fetal tissues from anywhere, and it's not really available. What is available for the last 30 years or so is ESCs and iPSCs, and the cells really have the potential to differentiate to any cell type in our body. However, there is a really huge deficit in the protocols because it's very hard to mimic what happens in real life in the bioenvironment, providing all the cues and all the factors and all the whatever the cell needs to differentiate properly with the right amplitude and the right timing. And what we do in Renewal Bio is we're not pushing the cells into going into a specific fate. We're just providing all of the bioenvironment that supports proper differentiation. How do we do that? Our vision is basically taking a cell from a biopsy from a patient or even a blood sample, reprogram the cells into iPSCs, transitioning them into one of our patented technology, which is the naive condition. And the naive culture conditions basically allows us not only to differentiate the cells to the embryo proper or the human body, but also the extraembryonic tissues that provide the support for proper differentiation. When we aggregate them together and we basically make the stembroids, we mature the stembroids up to a specific point and then basically collect the stembroids, associate them into a single cell suspension, and then we are isolating the cells that we need for whatever condition, and we transplant them back. And that's basically our vision. That could be used for a lot of different conditions, as I mentioned before, but there are a lot of organs that age, and we can rejuvenate them just by providing young, identical cells. And our platform is comprised of 3 breakthrough technologies. One is the ability, as I mentioned before, to culture our cells in naive conditions, allowing more plasticity and providing the supporting environment for proper differentiation. The second is the ability to grow embryos or stembroids outside of the uterus with our roller culture, ex utero incubator. And the third is basically making the developmental models from cells alone. So without a need of oocyte or sperm, we can take pluripotent stem cells, culture them in these conditions, make the stembroids, and basically make something that looks very much and mimics embryo development. It's not an embryo, it's just a platform to differentiate cells. How do we do that? So we take the human pluripotent stem cells and we take them from, so if we generate human iPSCs, the conventional ways to culture human iPSCs or ESCs are primed conditions. And our proprietary media conditions, the naive conditions, we take the primed condition, the primed cells, de-differentiate them back into the naive conditions, and from there we create the trophoblast, which would later, trophoblast cells, which will later form the placenta, and hypoblast cells, which are the primitive endoderm and extraembryonic mesoderm, more supportive environment that would later form the umbilical cord and the yolk sac. And when we aggregate all these cell types together with specific ratios and quantities, we form, after 8 days, what you can see here. So on the left panel, you can see, where's the pointer here? Is this it? Yeah. So on the left panel here, you can see a stembroid, 8 days in protocol. And on the right, you can see a textbook image of an embryo 14 days post-fertilization. And you can see that the structures here, so in magenta, you can see the trophoblast, as in here. And in yellow, you can see SOC17 positive cells, which mark the hypoblast or the yolk sac. And here, as you can see, is the epiblast. And what you see here is the bilaminar disc, which is a really important event in your life. Not your marriage, not your firstborn. This is probably the major event in your life. This is where differentiation happens. This is where gastrulation starts and elongates. And then later on, there's somatogenesis and organogenesis. So this is the proper way to differentiate cells. These are the conditions that allow proper differentiation into multiple and various cell types. So as it starts here at day 0, you can see a bunch of cells. At day 1, they start to cluster together. At day 2, day 3, they start forming compartments like the epiblast and the hypoblast. As they move forward, you can see that they grow in size, but it's kind of hard to see it in a bright field image, but you can see here by the staining that they form the proper, proper tissues and proper lineages. This is the second part. So first, before I showed you 2 parts of our technology, basically forming stembroids from cells alone, and also culturing in naive conditions. And this is the third part, basically growing. I don't know if this is, why is it not working? I'll try to get that here. So basically allowing embryos to culture them outside of the uterus. You can see here, this is a naturally mated, naturally conceived embryo that we took out, a mouse embryo that we took out at day 6.5, and we allowed it to grow outside of the uterus for another 6 days. And what you can see here is the elongation, the primitive streaks, and the somites forming next to it. So you can see that it not only growing in size, but actually differentiating and forming the proper organs and whatever is needed for proper differentiation. The beauty about our platform is that we're not pushing the cells into a specific fate. And as we move along with the axis of development, we see that we can use more and more cells for different indications and different conditions. And we're currently at this stage, day 23, 24, basically the onset of gastrulation, the elongation of gastrulation. And our first milestone is day 28, basically deriving hematopoietic stem cells. And there are a lot of evidence in the literature when you take young blood and you transfuse it into an old mouse, you can rejuvenate the tissue and basically prolong its life. And also with the allowing it to resist different pathogens more efficiently. But we can use a lot of different cells in each and every time point for a lot of different indications. So we're basically using the same platform, using the same protocol. We're not changing anything other than the way we're isolating the cells. This is basically a stembroid where we are now. A stembroid is day 23, mimicking 23-24 of development. You can see the hint gut here on the right. You can see human embryos at day 23-24. This is taken from atlases in basically available online. And you can see here the hint gut and the neural plate here and the amnion. And you can see these structures here as well in the stemroid. You can see the neural plate and the hint gut. There's the amnion and there's the trophoblast. So very exciting times. We can take these stemroids, dissociate them into single cells, and we magnetically separate specific cells. So we are now dealing with, at this stage, we're dealing with hematoendothelial progenitors, where CD34 cells can express both CD31 and CD90, basically indicating their dual lineage progression. And when we take these cells, we see that there are CD34, some of them are CD31 positive. They have properties for endothelial cells that we checked, and they are forming vessels. I think I showed you that last year. But what we see now is they're also expressing hematopoietic traits like CD34, of course, but also very important transcription factor. It's called RNX1. And what we've seen when we took these cells and we RNA sequenced them, something very interesting. We saw that if we try to cluster them with adult HSCs, and we could try to cluster them with data sets of human AGM, that's the area in the human body where, not only in the human body, but mammalian cells, where the hematopoietic stem cells generate. At first, so that's the first indication we're in the area or developmental stage where hematopoietic stem cells start to develop. We can see that our cells cluster with AGM and not adult HSCs, and we saw that they have both endothelial and hematopoietic traits with the Go ontology profile. So our vision, as I mentioned before, is to produce a lot of different cell types for a lot of different indications, and basically the process would be using either autologous iPSCs, allogenic iPSCs, or even engineered iPSCs, putting them in our platform, transition them to naive iPSCs, and in naive condition, allowing them to differentiate also to the extraembryonic tissues, aggregate them to stemroids, and then culture them up to the specific point where we want the cells, whatever cells that we're going to use, and making a master cell bank of a lot of different cell types that we can use for longevity or other conditions. And that's basically our vision and where we are striving to. This is the development pipeline. Currently, we're focusing on hematopoietic stem cells and endothelial cells, but as we go along, we can produce more and more cell types. We already see the starting of neuronal progenitor cells and a lot of different cell types that we can use in the future. So I would like to end with this. That's something for you, maybe, Ilya, more than anyone else. There are a lot of hubs in the world that are forming for basically progressing longevity treatments. So basically, after showing phase 1 safety, they're progressing straight away to humans. One of these places is Montana, but there are other places, the US and I think Panama and Norway and China in some places. So I was thinking, why not here? We can be pioneers in a lot of different things, and longevity, I think, is a really important thing that we can probably promote here in Israel as well. All right, thank you very much.
First of all, my name is not Harold, but if it was, I would be so happy to hand you a medal with the Alfred Nobel's face and $1 million check.
All right, come by our offices. We'll take a biopsy out of you.
So I would love to hear a bit about maybe the challenges that you encounter in developing the steroids and what goes wrong at the moment. So obviously
Sure.
That's a challenging environment, but How do you tackle that?
Have you tried it in your lab? Because that's a very specific thing. So there are a few challenges, of course. First of all is aggregating the right number of cells in the right, because now we're basically using a plate that has microwells, and you're basically seeding the number of cells, and you're basically counting on Gaussian distribution for the cells to drop in the well with the right ratio. And so that these are things that we tackle. First of all is the right amount and the correct amount of cells and ratio in each individual microwell. Each microwell is 400 microns. Basically from 1/6 plate we can make up to 35,000 stembroids. So that's one of the challenges, having correct and precise aggregation. Another thing is basically developing it further, and we're really relying on spatial organization. So for us, what goes well is when they're spatially organized, but the question is, what is the sweet spot of organization that you need in order to let the cells grow further? So what we see is basically we've spatially characterized up to 23, 24 days post-fertilization, but the structures don't die. They go further. We haven't characterized them fully in a way that I can say with confidence that, okay, these structures mimic embryonic development at day 28 or further. But we do get a repertoire of cells that we can use for a lot of different things. And now we're actually checking them in a single cell level to see, and also with a proof of concept and functional assays to see what they're actually capable of and how they compare to cells that have been, differentiated and directed differentiation, in vitro differentiation protocols. So now, basically, we're in a crucial point that we're seeing our cells, how they perform, and what's the potential. And something that is really important, unlike a lot of directed differentiation protocols, we've tried 14 different pluripotent stem cells, whether embryonic stem cells, autogenic iPSCs, and different iPSCs, 14 different cell lines, all of them produce stembroids. And basically the same success rates. So that's very encouraging because we see that our platform is very robust, and it is possible to translate it into autologous therapies. So in that aspect of development, these are the hurdles, but we also have hurdles with regulation, but we've been very proactive about it. So one of our advisors is a former cell therapy director in the FDA. And basically, we're aiming to take the same path, because it's a new entity, but we're trying to take the same path as cell therapy treatments. So hopefully this would pave the path for whatever other platforms. But we're actively working on putting it in the regulatory track. Thank you. Thank you.
Thank you very much. Thank you.